Every day we face hurdles in our life and problems that require new approaches or breakthrough innovations. In this podcast series, we're going to meet people that take audacious swings for the fence because they believe that changing the game is their personal responsibility. They believe it is up to them to be the solution. Hi, I'm Mark Forche. I'm the president and CEO of Delphinus, and welcome to our new episode of the Be the Solution podcast for dense breast screening. I have a fantastic guest for this episode. It's Dr. Mary Yamahsita, and Dr. Yamahsita is the Associate Professor of Radiology and Surgery at the Keck School of Medicine at the University of Southern California. And she is also the national PI of the Discover Breast ultrasound study that was done with SoftVue to support our PMA screening indication and all the work that fed those cases into the reader studies and ultimately the approval. Dr. Yamahsita is also now, post the approval, she is a consultant to Delphinus. So is it okay if I call you Mary?
Yes,
Yeah,
please.
okay, awesome.
Thanks.
Well, so listen, thank you for being here, and I'm really excited to talk with you because of this incredible role you've had in this innovation and bringing this innovation forward. So one of the things that jumps out to me as I think about this, so as you might guess for us as a company, having the national PI selected is a really key thing. And I remember I first met you in the Chicago Firehouse restaurant at the RSNA. You and Dr. Larson, and so you were both sitting at a table that at a presentation, and I sat down with you, and I was struck by your kind of enthusiasm for innovation and your interest in kind of challenges, and so what drew you to this as a project, and what kind of spurred your attention to this?
Mm-hmm. I was part of the Akron 6666 trial where we did whole breast ultrasound in high-risk patients. USC was part of that trial. And we saw that there was a need for breast ultrasound in patients because we were able to find additional cancers in patients by doing whole breast ultrasound. But at the time, we saw that it actually had too many false positives, too many unnecessary biopsies. And so it was not something that we were able to say, okay, I think everyone would benefit from ultrasound. But I knew there was a need for some type of ultrasound technology that would benefit women. And then later on, I was exposed to ABIS. I was actually part of their reader study, and I thought, wow, this is great. But I felt like there was something more that needed. And when I was invited to your dinner with the presentation, I thought, well, it's a brand new technology. I've never heard of SoftVue. I want to go and see what this is all about. And yeah, I remember being in that firehouse. It was really cool. And I remember Neb, one of the co-founders, he gave the presentation, and I thought, wow, this is so different. It's not just about looking at morphology and the grayscale ultrasound imaging, but there was something more, more of tissue characterization. And I just got really excited. Maybe this is something that would bring everything together, but it was such a new concept. And I was just really excited to be part of it.
Well, so, there's so many elements of the technology and what it means to you as a radiologist and to you as a clinician. But there's also, and I mean, you mentioned the other things too. There's kind of a workflow component too, that how do you integrate those things in to serve the, 40 to 70% of patients that could be dense breast patients, depending where you are in the world. And was there something in this that you looked at and you saw that also? I mean, that kind of the automated element did you see that offering advantages possibly too?
Yeah, so, with handheld ultrasound and even with ABIS, you still need a technologist. With handheld, it's operator dependent. And with SoftVue, it was going to be operator independent. Anyone
Yeah.
can be trained to
Yeah.
perform the study, and that would really help us as far as workflow, getting the mammogram and getting SoftVue at the same visit. It just sounded like, could this really happen?
Yeah. Well, so I was struck by your kind of curiosity and struck by your enthusiasm for taking on that challenge. But you know what? It's a pretty significant challenge too, because at the time that technology had been developed and had been put into use in some small studies, but this was a big study, and I think we're looking at 10 sites and 8,000 something plus patients, which is what we did ultimately. But at the very beginning, when you were looking at this, it was pretty much a blank sheet of paper. So, what kind of challenge did that pose for you as you thought about? I mean, that's an important leadership responsibility in leading into kind of the unknown. So, how did you think about that a bit?
Actually, for me, it was more exciting. For
Yeah.
me, it was a challenge that I really wanted to take on.
Yeah.
Brand new technology just seemed so amazing, and I really wanted to be part of it.
Yeah.
And I knew that I had a good team at USC. We have a really good research team in our Department of Radiology, and I knew that I would get the support I would need. So, yeah, I mean, I just took it as a challenge.
Yeah. So I recall. So we put a system in your facility, and everybody started being trained on how to actually screen the patients, and then and then read with the workstation. There's different sequences to go through, but and there was experience to refer to then, but then we're leading into the study where there was going to be a lot to learn. And there's always, with something like this where you have a breakthrough and you're about to go into this big effort, there's always a big decision point that you have to say, okay, let's go. So it's a point where you've got the regulatory things all in place, and you have to say, okay, when do we enroll patient number one in this? And your decision making in that was really important. So maybe just take me to that process and describe that a bit, what you were thinking and what was important to you leading into that decision.
Yeah, remember before we launched ARM 2, or ARM 1 actually, which was the prospective case collection study. Prior to that, we did ARM 2 where we actually enrolled patients that were scheduled to have breast biopsies. And so we were able to scan patients and then look at the findings and then correlate it to pathology. So we had benign lesions, we had malignant lesions, and then we were able to, I think, enroll about 500 patients. So during that time, we really needed to review all the images, come up with a reading protocol. But I think doing ARM 2 was really helpful because it was such a brand new technology and we're not used to interpreting images in a coronal plane. And then also having, 4 different sequences and looking and trying to figure out how do we differentiate breast tissue from masses and the masses from, is it benign, is it malignant? So I think during the ARM 2 phase, we really got to look at a lot of cases, biopsy proven cases. And so that kind of helped us to get organized, learn how to interpret images. And so once we got to a point where we felt like, okay, I think we're ready, But I remember asking you, is this the best technology we have available?
Yeah.
Are we ready?
Yeah.
Because we're going to continue to learn. I knew once we launched ARM1, it was going to be huge, but we were also continue to learn this new technology. So I remember asking you that question.
Yeah. Well, and that was, I mean, certainly that particular meeting for me is one of the most pivotal moments of my life. I think of all the things that all the people in the team had done to get us to that moment. And kind of the background that was really important that gave me the confidence to be able to say, yes, this is the time for the technology to move ahead, was we had had an intense internal effort for a long, long time to put everything on the table, from the best thoughts that we could possibly, kind of the most outrageous, audacious thoughts of how far can we push, distilled all those things down and put the things that we really knew made a difference into the system to get us to that point. So it was a big moment when you asked that question. It was important for me to be able to look in my rearview mirror internally and say, yeah, this is the very best that we can put on the field. And for me to say yes. And then that lets you say, okay,
let's do it.
and so, I mean, incredible moment to enroll that first patient, but also recognize, so that's just the beginning, and there were many learnings to come, because I know your team had done so much to understand the reading and the flow from the other work that had been done. But then you get 100 patients in, 200 patients in, 300 patients in. We were constantly learning things about the image interpretation, and constantly thinking about, okay, ultimately, when we're in the reader study, assembling all the best practices and all the things that we've learned. So maybe just tell me kind of the things that you learned in that time frame, and what that experience was like for you, because that's a very innovator oriented process and experience.
I mean, I learned so much during ARM1 while we were collecting cases. I don't feel like I ever stopped learning.
Yeah.
I kept learning because, with new technology, you have to try different things and say, okay, well, should we start with this sequence? Or, I mean, I think by then we knew how we were going to use the sequences, but just looking at different pathology and different findings, I was continuously learning. And for the prospective case collection study, we actually read mammogram first and then SoftVue, and we were able to incorporate SoftVue findings with the mammographic findings. So we were constantly learning. And I felt like, I mean, with any brand new technology, you learn. I remember when digital breast tomosynthesis first came out, there was a huge learning curve for everyone. So I went into this knowing that I was going to continue to learn. I didn't think that, oh yeah, I'm ready when we launched that very first patient. I knew that we would continue to learn. And I did.
Yeah.
And we all did. I shared, I remember having like conferences with other sites and just going through cases and learning from each other, learning from each other's cases. I thought that was so important.
Yeah.
Throughout this,
Yeah.
process.
And for me to observe that process and that kind of collection and assembly of best practices, I felt like we were really cutting new paths and finding new ground, because the images don't look like anything else. I mean, it's a very different process. there's 4 sequences. How do you manage them? How do you prioritize them? And I mean, you were super tenacious and diligent about doing that, and the entire team contributing. So I mean, I think those sessions were pretty amazing. But ultimately, so we got the cases we need to fill in the required things for the reader studies, and it tipped into a whole different thing, and that is taking people that had 0 experience with SoftVue and training them to go into a reader study setting. And I mean, our objective was to improve sensitivity and specificity. That's a big swing for the fence. We did. And so when you think about that training process and the things that went into that, so just tell me how do you assemble everything and get it into those modules that were done, because it was a pretty impressive thing.
I just remember spending, honestly, I think I spent thousands of hours looking at images, looking at our cases from our site. Because when we went from arm 2 to arm 1, we're looking for small cancers that are not seen on mammogram. So it was very different. I think with arm 2, initially, we saw bigger masses. And then when you're talking about screening population, you're talking about finding smaller cancers. And so that was different, and that was a learning process. And, learning the difference between a focal, stiff, dense tissue versus a small focal cancer. And so that was a learning process. And so for me, it was about just reviewing images over and over again, coming up with a reading protocol and making sure it works. Before I could confidently teach someone how to do something, I have to be confident.
Yeah.
So I just spent many hours looking at all those coronal slices, and I thought about what's the best way to teach someone how to read SoftVue. Well, breast imagers know how to read 1 millimeter slices because we are used to reading digital breast tomosynthesis, just going through slice by slice.
Yeah.
And then we're used to looking at different sequences when we do breast MRI. So I kind of brought those two together to interpret SoftVue, and I wanted to make it as simple as possible, because in order to read a study for a screening modality, it has to be fast. You can't spend 20 minutes interpreting a study. We spend about 2 to 3 minutes reading tomosynthesis. So I knew that with SoftVue, it had to be like that.
Yeah.
So just going through and just reviewing cases just over and over again. And at some point, it's like, this makes sense.
Yeah.
And then we came up with the lesion characteristics matrix. Talking about what you're supposed to see, the morphology, and what you're supposed to see with the different sequences. And then once we created it, remember we went through multiple versions before we finalized it, because I needed to go back and review all the cases again to make sure it really worked. So once we made sure that the summary of how to interpret SoftVue was formulated, and I felt confident that we had the best reading protocol. The teaching part wasn't hard for me, to be honest. We had radiologists during the reader study. We had radiologists that were right out of training, many years of experience, community setting, academic setting, but they all have the basic idea about reading, going through 1 millimeter slices, looking at different sequences. So the teaching part was, I felt like it wasn't that difficult once we came
Yeah.
up with a
Yeah.
with a final reading protocol.
Well, you know, I mean, the highest stage of learning is when you're unconsciously competent at what you do. You know, you don't even have to think about it. And I think what, at least from my perspective, what came from all this effort and all this understanding of dynamics of, you know, images and tissue flow and all the things that came out of it is the ability to just create a really deep understanding that actually winds up being pretty incredibly easily conveyed. And it's so logical that it makes a lot of sense. And so of course, then we, you know, that went into modules. People use that. They go into the reader study. They were challenged with things they'd never seen before, using new tools, and then they get this kind of audacious outcome of 20% increase in sensitivity and 8% increase in specificity. So now we're kind of on the other side of that, right? And so now that approval is achieved. So congratulations, because you took on this audacious challenge, and as a innovator and somebody doing something really incredible, you know, in your rearview mirror now you can see that, you know, leading the effort that got us there to that. So now, how do you think about the value of what that brings to changing the lives of dense breast patients and their screening? What do you think it means now?
Well, first of all, you know, once I trained the readers, now it was up to them
Yeah.
to do, you know,
Yeah.
I mean, I didn't know what the results were going to be.
Yeah.
I had no idea
Yeah.
what the results will
Yeah.
be.
Yeah.
And I mean, to show 20% sensitivity, increase in sensitivity is so amazing. But the important part is the increase in specificity.
Yeah.
Because up until now, breast ultrasound actually decreased specificity with increase in unnecessary biopsies.
Yeah.
And so when we got those results back, when I heard that, I was just so thrilled.
Yeah.
You know,
You think you were?
I'm sure. Yeah.
So
Yeah. I'm sure your company,
Yeah.
everybody on your team
Yeah.
were just
Yeah.
excited. And but just thinking about what this technology can do for patients, it's really exciting.
Yeah.
Because we have this need to do breast ultrasound in women with dense breast tissue. I think nobody would argue that.
Yeah.
But to not decrease specificity, I think that's just so important. And I'm really looking forward to seeing how this would play out in the clinical setting.
Yeah.
You know, but I think it's just wonderful that we can actually do screening mammogram and screening ultrasound on the same day.
Yeah.
And we can actually interpret mammogram and SoftVue together.
Yeah.
You know, a lot of times we'll see masses on mammogram, and if the patient's never had prior ultrasound, we have to bring the patient back and do ultrasound. But, you know, I could see, because, you know, we could differentiate cysts from solid masses. And if we see a bunch of cysts, patient doesn't need to come back for additional imaging. We could just call it BI-RADS 2.
Yeah.
And we're done.
Yeah.
You know, I mean, so there's potential to decrease callback rate.
Yeah.
For these unnecessary, you know, benign appearing things that we see on mammogram. So it'll be really interesting to see how this would play out clinically. But, you know, up until now, the ABIS, you can't do ABIS until you have a BI-RADS 1 or BI-RADS 2 mammogram, and then you do the ABIS, right?
Yeah.
That's how it was FDA approved.
Yeah.
So if you So the workflow would be, do you read the screening mammogram while the patient's there? So you determine if it's BI-RADS 1 or 2, and then patient goes and gets the ultrasound, the ABIS ultrasound, or do you just batch read the screening, and then bring patients back for automated whole breast ultrasound on another day?
Yeah.
But with SoftVue, the workflow, the way that it's been FDA approved, is that you can have mammogram and SoftVue on the same day, one after the other. As long as you have dense breast tissue, you can have SoftVue.
Yeah.
So as far as workflow, we could batch read these screening cases with SoftVue. Batch reading is so much more efficient, when it comes to interpreting screening technologies or screening procedures. And so I think it's really going to change the way we offer breast ultrasound to patients with dense breasts.
Yeah. Which is, I mean, that was the goal, right? So, so maybe just as sort of a final question here. So, it's pretty remarkable to have set sights on a goal with so many potential ways that something could go sideways and not achieve it, but then at the end of it to achieve it. So here we sit today with the outcome that was achieved. Rewind back to that moment, sitting in that room, making the decision about enrolling the first patient, and the challenges and uncertainties as an innovator. So how does this feel to have kind of climbed that mountain?
It's amazing. We went into this trial knowing that we would face a lot of challenges. With brand new technology, I would imagine there are all these challenges. But every time we came to a challenge, we were able to overcome it. We just kept going and going. And to get to this point where, to get these results from the reader study is amazing. But for me, I feel like this is the beginning. Now it's the beginning of what we can offer to women, and we're still going to learn so much. Because going from a reader study, clinical trial reader study, and then to clinical setting, there are going to be new challenges. So I think we're going to continue to learn, and with the goal that we will be able to really serve those women with dense breast tissue that need whole breast ultrasound.
Yeah. Well, look, thank you so much as the PI, as the leader for this effort, and also what you did to lead the many investigators and the other passionate people that participate in this. That'll be another podcast in the future, but thank you for the work that you did. And then just for the viewers, I'd like to say, this is really a perfect example of why we're doing this podcast. This podcast is about seeing challenges and taking them on with audacious thoughts and being the solution to those challenges. So I think we're all passionately committed to finding the ways to address the unmet needs of dense breast patients. And this entire effort was about exactly that. It was loaded with technology development challenges, understanding of new technologies and what it can do, and those challenges, the amazing tactical things of challenging patient enrollment and those tactical pieces, and then taking those findings and putting them into the right kind of setting to be able to demonstrate that. And that's what happened here. It is about being the solution when faced with all those things. Mary, thank you so much for being part of this and conveying all the things that you experienced and all the learnings and your just the things you experienced as an innovator. I really appreciate you being part of this.
Well, thank you for having me.
Yeah. Well, and to the viewers, thank you for joining us. Look forward to having you join us in the future. Please watch this space for new topics to come, and look forward to speaking with you again soon.